Good day. This video discussion will now be focusing on disaccharides, oligosaccharides, and polysaccharides. So we're done with monosaccharides, so we will now be going into the longer chains of carbohydrates. Okay, starting first with disaccharides. So, what are disaccharides? A disaccharide is a carbohydrate in which two monosaccharides are bonded together. In disaccharide formation, one of the monosaccharide reactants functions as a hemiacetal and the other functions as an alcohol. So, among the two monosaccharide components now that make up a disaccharide, one functions as a hemiacetal, whereas the other functions as an alcohol. Now, the bond that links the two monosaccharides of a disaccharide together is called a glycosidic linkage. So, diba, you have two monosaccharide units bonded to each other, man. So, the bond or the link between the two monosaccharide units is what we call a glycosidic linkage. Now, this section deals with the specific structures and properties of four important disaccharides. So, we will be dealing with some biochemically important disaccharides, namely the maltose, cellobiose, lactose, and sucrose. Starting first with the disaccharide maltose. From its very name, maltose, malt, often called malt sugar, is produced whenever the polysaccharide starch breaks down, as happens when plant seeds germinate and in human beings during starch digestion. So, in starch DAO, a very long chain of stored carbohydrates in plants is broken down, it results to the production of maltose. Now, maltose is a common ingredient in baby foods and is found in malted milk. Structurally, maltose is made up of two glucose units, D-glucose units, bonded together via a glycosidic linkage. So again, to make up the disaccharide maltose, you have to have two D-glucose units. Now, proceeding directly to cellobiose, the second disaccharide, cellobiose is produced as an intermediate in the hydrolysis of the polysaccharide cellulose. Like maltose, it is composed of two glucose units, the difference being in the way the two glucose are linked to one another. Another difference between maltose and cellobiose is that maltose is produced as an intermediate in the breakdown of starch, diba? Whereas cellobiose is produced as an intermediate in the breakdown of the polysaccharide cellulose. So again, maltose produced when starch is broken down, cellobiose produced when cellulose is broken down. Starch and cellulose are both polysaccharides. Now, like maltose, cellobiose also have two glucose units. They differ lang in the way these glucose units are bonded or linked to one another. Now, despite the similarities of maltose and cellobiose, they have different biochemical behaviors. Okay. Now, one striking difference between the two is that maltase, the enzyme that digests maltose, is found in the human body. Thus, we can easily digest maltose. Cellobiase, on the other hand, the enzyme needed to digest cellobiose, is absent in human digestive system. The cellobiose cannot be readily digested by humans. What does this mean? Now, to digest carbohydrates, we have to have the enzymes needed for its digestion. Okay? For maltose, we do have the enzyme needed for its digestion. It's called maltase, found in our human digestive system. So, kaya nato ma-digest si maltose because we have the necessary enzymes needed for its digestion. The opposite happens with that of cellobiose. We do not have the necessary enzyme called cellobiase, in the digestion of cellobios. Therefore, we human beings are not capable of digesting the disaccharide cellobios. So that is one of the um, striking differences between maltose and cellobios, who share a lot of similar characteristics. Okay, now going now to the next saccharide, we have here lactose, famously known as milk sugar, diba? Lactose is made up of one D-galactose unit and one D-glucose unit joined together again by a glycosidic linkage. So again, lactose is made up of one galactose, one glucose unit. Now, lactose is made up of, again, going back to that, one galactose and one glucose unit. Lactose is digested by the body using the enzyme lactase. So, the lactase is the enzyme needed for us to digest the disaccharide lactose. However, there are certain instances when a person lacks the enzyme lactase needed to digest lactose. When that happens, that leads to a condition famously known as lactose intolerance or lactose persistence, a condition wherein a person lacks the necessary enzymes needed to digest lactose. So, if you want to learn more on lactose intolerance or lactose persistence, you can refer to page 40 of your book. You have there Chemical Connections 1A, lactose intolerance or lactate persistence, the inability to digest lactose because of the lack of the enzyme lactase in a person's digestive system. Okay, last disaccharide is called sucrose, so commonly known as table sugar, the one we use on a day-to-day basis. It is the most abundant of all the disaccharides and occurs throughout the plant kingdom and is produced commercially from the juice of sugarcane and sugar beets. We are very familiar with this one, I believe. Tubo, locally known as tubo, the plant locally known as tubo is the main source of the disaccharide sucrose, which again we commonly use on a day-to-day basis as the table sugar. Now, sucrose, how is sucrose digested? Now, when we consume sucrose, it is broken down into equal parts glucose and fructose. That is because sucrose is made up of one glucose and one fructose unit. Now, glucose ultimately gets taken up by our cells with help of insulin, while fructose is handled in the liver and does not need insulin to be absorbed. So, again, what happens is that pag ma-breakdown mo gud sa sucrose, mag-result siya to the separation of its one glucose and one fructose unit, di ba? Now, glucose, since glucose is the main source of energy of our body cells, it is readily taken up by our cells with the help of the hormone insulin, whereas fructose will then be handled by another parts of the body for it does not need insulin to be absorbed. Regardless of its source, sucrose provides 4 calories per gram, and our bodies process it in similar ways. So, regardless of the source, sucrose always provides 4 calories per gram. Now, the enzyme needed to digest sucrose, what do you think is the name? Sucrase. Sucrose, sucrase, lactose, lactase. Ring a bell na sila yung similar na mga patterns, di ba? Now, despite the fact that regardless the source, sucrose provides 4 calories per gram, this is not to say that all sources of sucrose are nutritionally equivalent. Some sources provide more nutrient than others, which can affect how we metabolize sucrose. One such nutrient is fiber. While not always the case, fruits, vegetables, and nuts tend to contain more grams of fiber per calorie than most packaged foods and beverages with added sugars. Fiber helps to slow the rate of stomach emptying and digestion while also reducing glucose absorption. Thus, meals that contain fiber generally do not impact our blood sugar as much as meals without fiber. So, what I'm saying here is that there are some sources man good of sucrose that is more nutritional It's more nutritious than others because along with acquiring sucrose, naamo yung mga sources po that can give other nutrients aside from sucrose. Like for example, fiber. Now, fiber is a type of indigestible carbohydrate which possess a lot of benefits for the body. So aside from providing your body with sucrose, there are also other sources that can provide other nutrients as well. Therefore, um, it is good to acquire sucrose from sources that can provide you not just with sucrose but also with other types of nutrients as well. Now, as a disaccharide, again, sucrose is made up of one D-glucose unit and one D-fructose unit. I hope by now you have noticed that most of the monosaccharide units that we have met along our series of discussions only possesses the D-isomer of the monosaccharides, right? Again, that is due to the fact that the D-isomers of carbohydrates are the one that is most abundant in nature and is kwan pod kaning the one that is mostly used pod by different organisms. Okay, this slide has been nasaag this this has to be part of okay, our discussion for lactose. So going back, again, this slide is related to our discussion on lactose. It has been rearranged and it went all the way here. However, let's discuss why does milk turn sour? Now, again, milk is one of the sources of the disaccharide lactose, diba? Now, souring of milk is caused by the conversion of lactose to lactic acid by bacteria in the milk. Now, pasteurization of milk is a quick heating process that kills most of the bacteria and retards the souring process. Now, again, what causes the milk to turn sour? That is because of the disaccharide lactose being converted to lactic acid by bacteria present in the milk. Now, in order to address this problem, or in order to address to prevent rather to prevent this case from happening, pasteurization is done on the milk. Now, pasteurization is a type of process that kills the bacteria present in the milk, therefore preventing um, the souring of milk. Now, pasteurization, by the way, is named after the one who have discovered it, the French chemist named Louis Pasteur. Okay, so we're done with the disaccharides, the two monosaccharide carbohydrates. Let's now go to oligosaccharides. What are oligosaccharides? Now, oligosaccharides are carbohydrates that contain 3 to 10 monosaccharide units bonded to each other via glycosidic linkages. Again, when a carbohydrate is composed of 3 to 10 monosaccharide units linked to one another, that is an oligosaccharide. Now, two naturally occurring oligosaccharides are the trisaccharide raffinose and the tetrasaccharide stachyose. Now, again, for our discussion, we will be focusing on two naturally occurring na mga oligosaccharides. We have the trisaccharide raffinose and the tetrasaccharide stachyose. Trisaccharide composed of 3 monosaccharide units. Tetrasaccharide, 4 monosaccharide units. Now we have here the components of the oligosaccharide raffinose. We have one galactose, one glucose, and one fructose unit. Whereas the components of stachyose, the same to raffinose, but you have an additional galactose unit. Now, more on raffinose and stachyose. Now, humans lack the digestive enzymes necessary to metabolize either raffinose or stachyose. Hence, these oligosaccharides pass undigested into the large intestine, where bacteria act upon them, which usually produces discomfort and flatulence or gas. We do not have the capacity kuno to digest these oligosaccharides because, again, we do not possess the necessary digestive enzymes needed for their digestion. Therefore, these oligosaccharides will pass undigested along the intestinal tract, where sometimes they could be acted upon by certain types of bacteria, which will then result to certain na mga discomfort in our digestive system, like gas or flatulence. Now, an important aspect of human body chemistry that involves an oligosaccharide attachment to more complex molecules is blood chemistry associated with blood type. That is because blood types of humans is determined by the type of oligosaccharide present in or attached to red blood cells of certain human beings or kaning ginatest nga blood. The type of blood a person has is determined by the type of oligosaccharide that is attached to a person's red blood cells. So ang basihan daw kuno sa atong blood type is the type of oligosaccharide that is attached to a person's red blood cells. Now, if you want to learn more on this topic, you can refer to page 48 of your book, Chemical Connections 1D, Blood Types and Oligosaccharides. Okay. Now let's go to the longest of all the carbohydrates, the polysaccharides. Now, a polysaccharide is a polymer that contains many monosaccharide units bonded to each other via glycosidic linkages. They are also often called glycans. Okay, now how do we distinguish between polysaccharides? Again, polysaccharides are the longest chains of carbohydrates. They are made up of a lot of monosaccharide units bonded to one another. Now, how do we distinguish one type of polysaccharide from the other? Now, there are important parameters that distinguish various polysaccharides from each other. First, the identity of the monosaccharide repeating units in the polymer chain. Now remember, di ba, polysaccharide is a very long chain of carbohydrates composed of a lot of monosaccharide units bonded to one another. Now, for example, pag isa ra gani ka type of monosaccharide unit ang nagbalik-balik og kalink ana nga very long chain of polysaccharide, that is called or that would be referred to as a homopolysaccharide. Homopolysaccharide is a polysaccharide composed only of one monosaccharide repeating unit. For example, starch is a type of polysaccharide found in plants composed of a long chain of glucose repeating units. So taas ka siya nga chain of carbohydrates composed of daghan kaayo nga mga glucose units bonded to one another. Heteropolysaccharide, on the other hand, is a type of monosaccharide that is formed by two or more ng mga monosaccharide units. Now, another parameter that distinguish various polysaccharides is the length of the polymer chain. The length varies among polysaccharides. There are certain types of polysaccharides na dili kaayo long, whereas there are also types of polysaccharides that is very, very long. Now, some polysaccharides are much longer than others. Polysaccharide chain length can vary from less than 100 monomer units to over 50,000 monomer units. So the length of the chain varies between polysaccharides. Third parameter would be the type of glycosidic linkage between monomeric units. As with disaccharides, there are several different types of glycosidic linkages encountered in the structures of polysaccharides. Fourth parameter is the degree of branching of the polymer chain. Now, the ability to form branched chain structures distinguish polysaccharides from the other two types of biochemical polymers. That is because ang chain man gud nga ma-form sa polysaccharides dili necessary nga isa lang ka straight nga chain. There are instances nga na ay branching na mahitabo in the structure of these very long chains of monomeric units. So it could either be branched or unbranched. Okay, now let's take a look at this illustration first before time we proceed to the next slide. The first illustration at the top is an example of a homopolysaccharide unbranched, walay branch. So, unbranched homopolysaccharide. Now, nung homopolysaccharide siya, it is composed of only one type of monosaccharide unit repeated along the chain. The second one is an example of a branched homopolysaccharide. It is still a homopolysaccharide. It's just that it is already branched. Na yung mga branches, you can see on the illustration. Whereas the third example is an example of a an unbranched heteropolysaccharide. So you can see that the different colors indicate the presence of different types of monomeric units bonded to one another in this type of polysaccharide chain. Okay, now we have here two types of polysaccharides. We have storage polysaccharides and structural polysaccharides. The difference between them is very easy. Storage polysaccharides, these are polysaccharides that stores carbohydrates. For plants, it's in the form of starch. For humans and animals, it's in the form of glycogen. Structural polysaccharides, these are polysaccharides that um, help help us or contribute as a structural component in different organisms. Cellulose for plants. So you know that cellulose is an important aspect in plant cell wall. So cellulose is a type of structural polysaccharide. Chitin is found in arthropods, in crabs, shrimps, ilabit ang mga exoskeletons. That is one of the product of chitin as a structural na polysaccharide. So that ends my discussion on disaccharides, oligosaccharides, and polysaccharides. In case that you have any um, confusions or any questions that you would want to address me, you just send me a message. Okay? Thank you for listening and have a great day.